This week we're going to start talking about plant cells. So we're going to talk about the anatomy of a cell, types of plant cells, and how we generate new cells through the process of mitosis. So we're going to start off with the anatomy of plant cells. So the objective here is to understand just cellular structure of the generic plant cell. What does it look like? What are the organelles? What are their functions? Keeping in mind that when I say organelle, it's essentially an organ. But rather than an organ for the whole organism, it's an organ in the cell. So it's like a miniature organ for a cell. So what is a cell? It's the smallest functional unit of life. All living things are made up of cells. There's no living thing that doesn't have a cell. Some organisms are single cells, so they consist of only one cell. Other ones consist of a lot. Plants are multicellular, so they have lots of them. But all living things are made up of it. It's the building block of life. That's what we call cell theory. So what's an organelle? It's basically an organ. It's an organ for a cell. It has a specific function within the cell. It performs a job that keeps the cell going. They have a bunch of different ones to it, but they all have their own individual job. So here's a diagram of plant and animal cells. The left is an animal cell. The right is a plant cell. You can see there's a bunch of organelles, and we're going to go through them one by one, so don't worry about writing them all down right there. But you can see right off the bat that they look very different. The plant cell is square because it has a cell wall. Animal cells don't have a cell wall. Plant cells have these chloroplasts because they undergo photosynthesis, and animal cells don't have those. Since they have a cell wall, they have other things like plasmodesmata, which allows them to connect to adjacent cells. And so there are quite a bit of differences between an animal cell and a plant cell. So starting off with the cell wall. The cell wall is that square structure in the green right here. You can see that. That gives support to the cell. It allows the the plant to build up. It has like basically bricks, good structural support, and it gives it protection. It also is going to prevent it from bursting. Now, we haven't really talked about diffusion yet, but if you put a cell, or really anything that's permeable to water, meaning water can pass through it, into fresh water, meaning water without very many solutes in it, so just fresh water, water will move into it. So water will move into the cell. If water is moving into the cell, it can burst. This is what can happen with animal cells. If there's not a mechanism to prevent it, the cell can burst in fresh water. Since plants have a cell wall around it, it's not going to burst because it's going to push against that cell wall, and the cell wall is going to stop it from being able to burst. So they're not really affected by freshwater solutions that might cause bursting in other cells. Now, if you look here at this picture, this is a close-up of plant tissue. You can see there's all of these cells here. These are cell walls. Now, these cells are really closely packed together. And to facilitate that, there's this thing called the middle lamella, which connects two cells together. It basically acts like a glue or cement to hold them together, and it allows them to adhere to one another and communicate back and forth. So the cell wall protects it. The middle lamella joins cell walls and allows for continuous contact, adhesion, and some communication as well. Then we have the cell membrane. So just inside of the cell wall here is the cell membrane. So it's hard to see in the diagram here, but there's a cell membrane around it. And the membrane is just this combination of phospholipids. So phospholipid meaning lipid with a phosphate on it. So you can see zoom in here of the cell membrane. These red pieces here are the phosphates. These little tails here are the lipids, so we call those fatty acid tails. And notice, so this red one and these two little legs here, that's one phospholipid. So here's one layer of phospholipid, and here is another layer of phospholipids, but they're in opposite directions. So we call this the phospholipid bilayer. And what this allows it to do is control what goes in and out of the cell. So it's what we call a selectively permeable membrane. It selects what should come in and out of the cell freely. Now the tails, since they're fats, they're hydrophobic. Fats and water don't mix, so they repel things like water. The phosphate part are hydrophilic. They like water. This hydrophilic, hydrophobic, hydrophilic contrast across the cell membrane creates a circumstance where not everything can go across. So, what can go across? Non-polar molecules. So molecules that don't have a charge across the molecule itself, they can go across. So stuff like glucose can go across relatively easy because it's an injury source for us. And then things that are really small can go across. Stuff like potassium or sodium or different things like that. Things that can go across that are really small or non-polar can get across. But how do the other things get in? Well, it's not shown here in the diagram, but there's actually proteins inside of the cell wall that we can either control, allow them to flow into the cell freely through these protein channels, or we can pump them across. And we'll get more into this later in the semester. But the cell membrane is there to act as a gatekeeper. What can go in and what can go out of the cell. Now, inside of the cell is a bunch of liquid, right? That's called the cytoplasm. It's just a liquid in the cell. It allows things to move through freely. It acts as like the solution of the cell. Now, in the cell wall, you can see these. We have several channels in here, right? Those are called plasmodesmata. Since there's a cell wall, it's harder to communicate with the adjacent cells. In our cells, it's really easy because we don't have any cell walls. It's just cell membranes. So they have cell walls. They have other mechanisms to allow for transport and communication between cells. And they do that by putting a hole in their cell wall called the plasmodesmata. It's basically just a channel that allows plants to send stuff back and forth to each other and to communicate. This will be really important when we talk about like vascular tissues of plants. If you recall, the vascular tissue in plants is composed of xylem and phloem. Now, the xylem is dead at maturity, so there's not much communication, but there is transport between the two adjacent cells. But with phloem, we have two types of cells that are really important. The sieve tube, which we haven't talked about yet, but just to demonstrate the importance of plasmodesmata, which facilitates the flow of nutrients and water. It acts as like a straw. And associated with it is what we call the companion cell. The companion cell is going to increase or decrease the pressure inside the sieve tube. And it does that through a plasmodesmata channel. Then we have the cytoskeleton. The cytoskeleton is the structural components of the inside of the cell. It's composed of microtubules, interfilaments, microfilaments, all kinds of different things that will allow this to move things in the cell around, split the cell during mitosis, and give overall intracellular support. So it acts as a transport system in the cell, it allows for movement in the cell, and it helps with the shape. Then we have the nucleus. This is the big part, right? This is the control center of the cell. It's this big purple part here, this big circle, right? That's the nucleus. It houses the genetic material of the cell. So that's where the DNA is. And you can kind of see inside there trying to note the little lines with this DNA. So it's the control center of the cell. It's going to regulate cell growth, how quickly the cell grows and how it grows. It's going to regulate metabolism, so how quickly it goes through cellular respiration, and reproduction, so how often it goes through mitosis. It's the control center of the cell and where DNA is found. It's also where ribosomes are made. Then we have plastids. Plastids is actually a group of organelles. So it's a group of organelles. We're going to talk about several different groups in them that are responsible for storage, for photosynthesis, for pigment storage, for a variety of different functions. But plastids is just a catch-all group for several different types. The most common one being chloroplast. The plast at the end denotes that it's a plastid. So chloroplast means plastid with chlorophyll, right? So it means it's the one that's carrying out photosynthesis. So chloroplasts, which you can see here, these green ones, very similar in shape to these mitochondria here, right? Or the plastids for photosynthesis, the chloroplasts. They're going to absorb light and produce chemical energy in the form of sugar. So they're taking light in, and they're going to make sugars out of it. So it's basically, in some context, the opposite of a mitochondria. Mitochondria is going to take in sugar and produce energy. The chloroplast is taking in energy and producing sugar. Then we have the chromoplasts. These are responsible for storing pigments. So the flowers, colorful petals of a flower, the color of a tomato or a fruit or something like that. Those are from the chromoplasts. And here you can see a tomato. And all of these red dots here are from the chromoplasts inside the cells, giving the tomato that red color. Then we have leucoplasts, which are responsible for storage. So they store starch or lipids or proteins, whatever it might be. Here, this is a type of amyloplast, which is a type of chloroplast found in a potato for storing starch, which is an energy storage molecule for plants. So starch dyes dark blue to black in the presence of iodine. And so these potato cells here have been dyed with iodine, and you can see very clearly the leucoplasts inside the cells. So here's one cell, and then here are the individual leucoplasts that have been dyed. Next, we have the mitochondria. Mitochondria is the powerhouse of the cell. So this is where we're getting energy in the form of ATP. Cells use ATP as the currency. So ATP just means adenosine triphosphate, and we're going to use those as the batteries to power different cellular functions. And mitochondria produce ATP by taking in sugars, digesting them, breaking them down, and ultimately making ATP. We'll talk a lot more about this process when we get to plant metabolism, but for now, just know it's the powerhouse of the cell. Now, what's also important to note is if you look at the chloroplast here and the mitochondria, they're about the same size. They have similar-ish structures, and they're also about the same size as a prokaryote, a bacteria or a archaea. What's also interesting is that they also have their own DNA. They have separate DNA from the actual host organism. It's like that because of what we call endosymbiosis. A long time ago, during the evolutionary history of animals and plants and eukaryotes, for whatever reason, a prokaryote was engulfed inside of a eukaryote and survived. And as a result of that, and this happened several different times, one for mitochondria, one for chloroplasts, and several different times in other organisms as well. We basically have, and plants basically have, prokaryotes living inside their cells. Mitochondria and chloroplasts are basically like bacteria that we co-opted to perform cellular functions for us. That's what we call endosymbiosis theory. That's why they're the size of prokaryotes, that's why they have their own DNA, and that's why they act a little bit different from the other organelles. And I'll post a video related to that. Then we have ribosomes. So ribosomes in this image are really kind of hard to see. They're really small in there. You can kind of see them there. And they're also studded across this rough endoplasmic reticulum. They're making proteins. So DNA is actually a code for proteins. That's what that's the function of DNA. But we don't want DNA to exit the nucleus because then it's going to be degraded and potentially harmed. And we don't want that. We want to protect our DNA. So we take our DNA, we transcribe it into mRNA, which we'll talk about later on. That mRNA then leaves the nucleus because we don't care about that quite so much. And it's going to go to a ribosome. The ribosome is going to read that mRNA, and it's going to make proteins out of it. So ribosomes are the protein synthesis organelles. Now, ribosomes are found in the cytoplasm or on the rough endoplasmic reticulum. That's why you can see the rough endoplasmic reticulum, or rough ER, looks studded. It has these little ribosomes attached to it. There's a reason for that. The rough ER is responsible for folding and manipulating the protein after the ribosomes make it. So proteins are really complex. We're not going to get into the detail of it, but they're folded in really complex patterns that are responsible for their unique chemistry. Now, the ribosome is going to make the protein, and then the rough endoplasmic reticulum is going to fold it and change it. On the contrary, the smooth endoplasmic reticulum, which you can see over here, is blue stuff, doesn't have ribosomes on it. And it's responsible for synthesis of lipids, which are fats, detoxifying chemicals, so removing chemicals that might be harmful to the cell, and not responsible for the protein pathway. Then we have the Golgi apparatus. The Golgi apparatus is basically like the USPS or post office of the cell. It's going to take in packages and ship packages. So they can take in, say, you have the ribosome that makes the protein. The rough ER is going to fold the protein, right? It's going to send it to the Golgi apparatus. It's going to package it and distribute it where it needs to go. It will do this with other stuff as well. It's not just proteins, but it's responsible for taking in stuff, packaging it, and shipping it. It could ship it to somewhere in the cell or outside of the cell, which is unique about it. So it's the distribution center, the USPS of the cell. Then we have the peroxisomes. So the peroxisome you can see right here. Its job is to break down fatty acid and detoxify harmful stuff. Now, it should sound a little bit like peroxide to you. So peroxide just something like this. I just happen to have it by me because I use it to clean some plant tools. But peroxide is harmful. It is not, it's basically a poison. We use it to clean wounds and stuff, but we produce it also in our bodies and we want to get rid of it. It's not necessarily the safest substance for a living organism. And it actually is produced by a lot of reactions that happen in our bodies and in plant bodies. So what peroxisome does is it actually is a little house to carry out those reactions. So reactions, chemical reactions that might produce peroxide will happen in the peroxisome. That way it can protect the cell from it, break down that peroxide rather quickly, and not let it harm the other parts of the cell. So it's responsible for detoxing those harmful substances. Now, this is one that's unique to plants and not an animal cell is the central vacuole. You see this big vacuole here. Now, vacuole just basically means like a membrane-bound container of some sort. It can be for storage or for storing nutrients or storing waste. The central vacuole, though, has a very important role. It's basically a big water-filled sac. So as plants absorb water, the water will move into the cell and it's going to be stored in the central vacuole. Now imagine what would happen if this central vacuole collapsed. If it lost all its water. The central vacuole disappears, all these organelles flow into the middle, and the cell wall starts to collapse. It collapses in on itself, right? That's called wilting. So the central vacuole is responsible for maintaining turgor pressure. As the plant absorbs water, it funnels it into the central vacuole, it blows up like a balloon, and keeps the plant cell really rigid or turgid. So the turgor pressure is maintained by the central vacuole pushing as it fills up with water against the cell to hold it nice and sturdy. So it's responsible for maintaining cell pressure or turgor pressure. Okay, so just to recap, we went through the cell structure, including all of the organelles. You can see the organelles listed here. So make sure you can identify all of those, you know their functions, and the general idea of what their role is and anything unique attributes about them.